over the muscle fiber.
She's gonna use that.
Here. Sorry.
No, you're fine. I'm just. No, I've never recorded her, but I think it's gonna be
Okay. Now, did I have all those notes and everything on the board for you last Friday about the sarcolemma and the sarcoplasm?
You did.
And myofibrils and all that stuff. Okay. So just remember that this model actually represents a very short segment of a very, very long skeletal muscle cell. Okay? And since it is so long, we tend to call them muscle fibers, right? These muscle fibers will be extremely long, and they will generally run quite often the length of the entire muscle. So in this sartorius muscle, those individual muscle fibers may be 12 inches long. That's a cell that's 12 inches long. Okay? So we just got a little bitty short representative segment. Now, I'm going to start with the exterior and work internal. We'll come back over here and talk about the neuromuscular junction and relate this to this drawing that y'all have. Everybody got one of these, right? If you didn't, you need to get one. Okay. So let's start off by identifying what this connective tissue membrane is that's around this muscle fiber.
Is that the sarcolemma or no?
Endomysium. Endomysium. Did we label it as such?
We did.
Okay. Endomysium is one of the 3 connective tissue membranes that compartmentalize all muscle tissue, the skeletal muscle tissue. So every muscle fiber will have a layer of connective tissue on it that's covering it. Kind of think of it like the insulation on the outside of a wire. Does that help? Because we're dealing with electrical signals, aren't we? All right? Insulation. Okay. Now this right here represents the plasma membrane of this cell, but we have a special name for it. What's it called?
Sarcoplasmic
Sarcolemma. Sarcolemma is the same thing as the plasma membrane. It has its own particular name, sarcolemma. What does sarco mean? Did I tell you what it meant? It means flesh. So, if you consume flesh of a chicken or a cow or a pig, are you eating meat? Are you eating muscle? Sarco means flesh, okay? So the sarcolemma is the plasma membrane covering the, defining the margins of the muscle fiber.
And the first one, what you said was the endo what?
Endomysium. And there are 2 other membranes that are discussed. We discussed them in lecture. Okay, what is this structure right here? It's basically an axon branch. This is an axon of what kind of neuron? A motor neuron. It's called a somatic motor neuron. It's part of the somatic nervous system that regulates skeletal muscle, okay? Now, these originate in the spinal cord. The cell bodies are in the spinal cord, and then they emerge, these axons emerge from the spinal cord by way of the spinal nerves. And they travel from there towards the muscles. So those axons can be very, very long. They can be up to 3 feet long. So as they start approaching the muscles, they start branching and branching and branching and branching, because that one neuron is going to synapse with many, many muscle fibers, okay? So this is just one branch of the axon, and these structures right in here, I refer to them as the terminal bulbs of that neuron, okay? That's the part of the axon that's going to form a synapse with your muscle cell. Did I give you a definition for synapse? Okay, it's just a junction between a neuron and another cell. It doesn't have to be muscle. It can be another neuron, it can be a receptor, some other type of cell. But this is specifically a synapse that's between a neuron and a muscle fiber, so it's called a neuromuscular junction.
So do you want us to put synapse right there? Or do you want us to put terminal?
I mean, you should know what a synapse is. Okay, this is just one type of synapse. But the combination of both of these areas right here would be a neuromuscular junction, wouldn't it? Okay. Now, do all cells have cytoplasm? Cytoplasm consists of a fluid media plus all the internal cellular structures, right? So what do we call the cytoplasm of a muscle
The
cell?
sarcoplasmic reticulum.
Sarcoplasm. Now the fluid portion of it, we can label as sarcoplasm, can't we? And that would be anywhere internally that has this light pink. We see some right here. Okay, so that would be specifically the sarcoplasm. Do our muscle cells have a nucleus? Do they have just one nucleus? They have multinuclei. And where do we see them?
The dark spots.
Are they in the center of the cell like we normally see? No, they're around the perimeter, aren't they? So we saw a nucleus right here. We saw one right here. There's one on the back side of that muscle fiber model. And then I think we see a couple in here. I don't know what else those would be if they're not nuclei. Okay? But they're not interior per se, are they? They're around the perimeter. You can see them on the outside of the cell.
They're in the sarcoplasm?
Yes, they are. But they're pushed out around the perimeter. Okay?
And what was pushed out? The nucleus?
The nuclei are not in the center of the cell. They are basically, the cell is filled up with something else. It's called myofibrils. So the only place for these nuclei to be visible is around the perimeter.
Okay.
So what you should see in the middle of this cell is a whole bunch of what are called myofibrils. Okay?
The straw.
The straws, yes. That's the best analogy I can always use. Now, these are going to run the length of the muscle too, aren't they?
Mm-hmm.
Yeah. And you're going to have many, many, many myofibrils. Would you estimate how many might be in this one little muscle cell?
10,000.
Could be 1,000. I don't know. It's going to be a lot. But anyway, each one of these myofibrils is made up of many, many repeating units that are functional. These are the sarcomeres. Do you remember the sarcomeres are the functional units of skeletal muscle? Okay. So, can you see these dark pink and light pink bands? Okay. Those dark pink and light pink bands and the black lines that we see separating them, these are telling you the beginning and the end of each sarcomere. Okay? So, on this little segment, I would say maybe each one of those myofibrils has 10 to 12 sarcomeres. Is that approximate?
Yeah.
So how many sarcomeres do you think are in every myofibril?
Those are tens of
A
thousands.
whole bunch. So the inside of the muscle cell is filled with thousands and thousands and thousands of sarcomeres, each of which can contract a little bit. Are y'all following me?
Yep.
Okay. All right. So let's look here at our color bands. What was the term for those color bands that we saw in skeletal and cardiac muscle?
Were they some kind of filament?
They are filaments, but we said that cardiac
Myosin.
and skeletal muscle look like it's banded. We call it striations, remember? So we have striations in skeletal muscle, and that's because we have two primary proteins that are used to make up our sarcomeres and our myofibrils. What are those two proteins?
Actin, myosin.
Actin and myosin. Which one is the thick protein?
Myosin.
Myosin. And what color is it in our sarcomeres here? The dark pink band, right? And that dark pink band, which is made up of myosin, what structure anchors those myosin fibers in place? The M line. Does it look like a little bitty short black line? Okay, that's the M line. That represents little filaments that are holding that myosin filament in place. It's anchored there. All right? Now, in these light pink bands, what protein is present there? Actin. It's a thinner filament, right? So it shows up as being lighter. Now, that light pink band would be called the I band, if you're looking at the sarcomere, right? The dark pink band would be called what band? The A band. And what proteins are present there?
Both.
Both of them, but primarily myosin, right? But actin does overlap a little bit with the myosin. So, what structure are the actin filaments attached to?
Z band.
Z discs.
Z disc.
Okay. Z discs. Now, if you're looking in the A band, we have myosin and actin overlapping, right? Are the actin filaments touching each other when it's relaxed? No. Is there a gap between them?
Yes.
What do we call that gap?
The H zone.
And that is how much that sarcomere can contract, right? That gap can close. Now tell me, do my myosin filaments change in length?
No.
Do my actin filaments change in length?
No.
Do the actin filaments get longer or shorter? No. So how does that gap close?
Through your H zone, right? Or it's the distance
overlap each
between.
What happens? Do they slide past each
Yes.
other? Yes, that's what's happening. It's called the sliding filament model. So, if you get a contraction, myosin reaches up and starts pulling the actin on both sides, right? So it closes the H zone. Okay? Do you have it? Understand? Is it time for a break? It's only 10:30. Okay. All right. Now, I can discuss most of what we just discussed and apply it to this picture, can't I? So, and you have this. You have it labeled. You also have descriptions of each of these features that are explained to you here. Did y'all color this? You didn't? We colored it different colors on the board, didn't we? Did I draw it on the board? Okay, I used pink and blue or something for my actin and myosin. Okay, so that's one good way of, if you color it, it's a process of learning. You know that that particular color is associated with actin, that particular color is associated with myosin, okay? All right, now if you haven't looked at the practice practical yet, this diagram, unlabeled, is back there. I've got all the different features labeled with letters. So you should be able to label this with the terms that we described. It's going to be a whole lot easier for me to ask you about features of the sarcomere from this than from this, isn't it? So, on this, it's going to be primarily superficial structures. I could have a sticker on this, or this, or this. I could ask you what these little brown granules are. Did we discuss that?
Mitochondria.
They're the mitochondria. Why do we have so many?
For energy.
You have lots of energy for contraction, don't you? Okay. Did we get this covered except for the synapse? Okay.
What about the little squiggly white lines and stuff on that?
Okay, that's what we're fixing to do.
Okay.
The synapse. That's the synapse. Okay. Now, just remember that a synapse, when you hear that word, it's a neuron that is forming a junction with some other cell. In this particular instance, we have a neuron, it's a motor neuron, and a muscle fiber. Okay, so this is the top picture. We're able to see more details about the neuromuscular junction. Okay, now, this membrane that's on the outside of the muscle fiber, what is that called? What is the membrane on the outside of the muscle fiber? sarcolemma. Sarcolemma. Okay? So sarcolemma covers the whole outside of the cell. So this is sarcolemma, this is actually sarcolemma, this is sarcolemma, and we continue sarcolemma. So the membrane on the outside of the muscle cell is called the sarcolemma. Okay? Now this is that terminal bulb. There are two of them here of the axon. They're coming very, very close to the sarcolemma, but are they actually touching? No. There's a little minute gap between them. And what is that little minute gap called? The synaptic cleft. Okay. So all of this is the sarcolemma. Okay?
I thought the endomycin was the most exterior part of it.
It's not the membrane though. And
So-
see, this is not showing the membrane right here.
So the terminal bulb is gonna be past the endomycin, then
Uh-huh.
you're saying?
Yes. Okay, so this is our axon. This is a motor neuron. So the axon's coming towards the muscle fiber. The signal will come from the spinal cord towards our muscle fiber. This is my terminal bulb of the axon. And this part of the membrane on that axon is called your presynaptic terminal. Presynaptic terminal. That's the membrane on the axon. The membrane over here, which is part of the sarcolemma, is called a postsynaptic membrane. Okay? This is how the signal is coming, presynaptic. It will relay that information to the muscle fiber at the postsynaptic membrane. Okay. Now, at the presynaptic terminal, this is where we can have the release of neurotransmitters by the axon. If the signal is strong enough, if it reaches a threshold intensity, it will cause the release of neurotransmitters that are in these little vesicles. And it releases them by exocytosis. Y'all should remember what that means. So if those neurotransmitters are released into the synaptic cleft, they come across and bind with receptors on that postsynaptic membrane. That area where the receptors are found is referred to as the motor end plate. Okay? The motor end plate is basically part of the postsynaptic membrane. And we identified the motor end plate as that white wavy line, didn't we? Okay? So, receptors will bind with the neurotransmitter, and that initiates a whole chain of reactions that results in contraction of the whole muscle. So, do you know what the neurotransmitter is?
Acetylcholine.
Acetylcholine, and how do we abbreviate that?
ACH.
Yes. Here it is right here. That is your neurotransmitter that will stimulate a muscle contraction. We have other neurotransmitters that are often hormones, but they don't necessarily act on skeletal muscle. They'll work on cardiac muscle or smooth muscle. So we'll talk about those a little bit when we finish up that chapter, okay? Now, let's transfer what we know about this to the picture, okay? So we know this is our axon, this is a terminal bulb, and the membrane of that terminal bulb is called our presynaptic terminal, right? So we identify the presynaptic terminal as this blue-gray line. That's the membrane. Do I have it here too? Yes. So that is where neurotransmitters can be released, right here. And could we find the synaptic cleft there? There's a little bitty gap. You could put your fingernail in it. So there is a cleft there. And then that neurotransmitter can come over and synapse or bind with the receptors on the motor end plate. The motor end plate is that white wavy line. So that's where we have the receptors.
So the postsynaptic membrane is basically the sarcolemma?
Yes, yes. And the motor end plate is basically part of it in that location. So we know that this is embedded in the sarcolemma, which is, in that region, the postsynaptic membrane. Okay? All right. Now we have sarcolemma everywhere, but this particular area of sarcolemma has the receptors in it. So it does have that motor in play, or that collection of receptors. So that is the postsynaptic membrane of the muscle fiber. All right? Was that everything? Are you getting it organized? Okay, good. Any questions about the muscle fiber? What about our little picture here? Tell me what defines the ends of your sarcomeres. The Z disc, okay? And what protein fiber is directly attached to the Z disc? Actin. Do I have actin from one sarcomere here and actin from another sarcomere over here? So just know these sarcomeres are joined together end to end, and you may have thousands and thousands of them in any myofibril. And then, of course, we have many, many, many myofibrils in each muscle fiber, right? So we've got these similar words, but you have to be able to differentiate what they are. Actin and myosin are called myofilaments. Myofilaments. You have myofibrils. The muscle cell is called a muscle fiber. So we've got several words there that are very, very similar. So don't get confused on them. Does that make sense? The myofilaments are actually actin and myosin. And you should know that one is thick, one is thin, and actin is bonded to the Z disc. Myosin is held in place in the sarcomere by the M line. Okay? Myosin is thicker, so it's going to look darker. It's going to form these striations. Any other questions? What do you need help with? Do you need to just spend some more time with the muscle girl? Do you need to spend some more time looking at those pictures that I provided on Moodle? You need to have both of those facial muscle pictures labeled. Okay? And I provided you labeled pictures by Moodle, didn't I? Okay. Now, what you're going to find when you look at the practice practical is that I've used muscle girls. I've also used the torso models. I've also used the facial pictures, but I've also used a couple of other pictures that look very similar to the labeled pictures you have on Moodle. I think they're really good, clear pictures. So you shouldn't have any trouble identifying the muscles. Have y'all looked at that back there yet?
Yes.
Okay, so there are a few pictures. Do you have a problem with the pictures? Good. And then, of course, I've got the muscle fiber back there. And I have this figure with the features labeled as A, B, C, D, E, F, right? Okay. So if you feel like you need to get some more pictures of the torso model or the muscle fiber or whatever, we've got plenty of time for you to get that today. We have another whole class period. So if you want to take a break, go get you something to drink, go to the bathroom, come back.